Measurement standards. I'm Ingrid Swanson, and I'm affiliated with Herbert Sarrow's lab, although I'm not technically a member of his lab. I run the iGEM team at the University of Washington. At the University of Washington this past April, we hosted the Standards and Specifications in Synthetic Biology conference. One of the things to come out of that conference was that we would host a session at SB4 about measurement standards, and that's this session. So I'm just going to talk for a few minutes and just get you thinking about measurement standards, and then turn it over to more of the experts in this field. And in total, we're only going to talk for about half the time, and then the other half of the time is for discussion, because these sessions are supposed to facilitate transfer of ideas about these topics. Synthetic biology is commonly compared to engineering disciplines, and engineering disciplines are very fond of standards. Well-established standards. For example, in synthetic biology, we have physical standards, naming standards, description standards, many of which were talked about in the last session. It's also important to define a biological unit in terms of its biological properties. And this is really only meaningful to us as synthetic biologists if you can compare it to some reference standard. And of course, the ultimate goal being that you can generate a system that has demonstrates some predicted desired behavior, and that you can do this in an efficient and predictable and reliable manner. So the question that is the focus of today's session is, can we implement meaningful measurement standards for biology? There are a lot of obvious advantages for generating measurement standards. I have three up here, but by no means this is it for advantages. Abstraction. If you have a biological unit that's defined in terms of its biological activity, then you can consider its activity in terms of input to output, and this abrogates the need to delve into the molecular processes that underlie this. Predictability. If you have biological parts that have had their activity characterized compared to some reference standard, then you should be able to predict how they will interact when you interface them together. And this should be true whether the part was measured in one lab or another lab, because if they're all using the same measurement reference standard, they should behave the same no matter what lab they were originally measured in. Choice limitation. Currently in the registry, there are hundreds of, for example, promoter parts. And I know for our iGEM team, at least, it's been difficult to select which parts to use for their circuit. And for example, if you were to undertake, say, some large metabolic engineering project, and you do some kind of modeling that tells you that to get the optimal flux through your system, you need promoter strengths that are certain levels. Having a library of well-defined promoters that have been compared to a reference standard will make it easier for you to select the parts that you want to use to generate your desired system. So we need standards, but a question that came up at the workshop was that which types of biological parts should be standardized, and with what accuracy can we do this standardization? Today we will talk about promoters and ribosome binding sites, standardizing these. Should we bother standardizing terminators? Would it be useful to us to standardize reporters? There are a number of different GFPs and RFPs and other reporters in the registry, and it could be useful to compare their intensities. And also another thing that we talked about at the workshop was what are the benefits of using absolute units to define the activities of these parts in terms of some absolute biological unit, or if it's more useful to describe them in relative units compared to a reference standard. So aside from these questions, there are other complications that are specific to synthetic biology measurement standards, because synthetic biology is a biological discipline. One of this is that to accurately measure biological activities, this is sometimes very costly and time-consuming and labor-expensive, or it can be. So because of this, it's sometimes difficult to convince and encourage labs to establish measurement standards for their parts. And we want to encourage labs to characterize and document their parts so that they can be used by other groups. Biological systems tend to be noisy, so you can have a group of cells that are genetically identical, but they can be heterogeneous in their gene expression. Synthetic circuits tend to be very context dependent, so they could behave differently depending on what strain they're in, depending on the growth phase of that strain, temperature, constitution of the media. And they also tend to be dependent on the sequence of the DNA that surrounds it. For example, ribosome binding sites may function differently based on the open reading frame that follows them. So if you have a transcript, the sequence of the ribosome binding site may be involved in some secondary structure interactions with the open reading frame sequence, and it would then modulate the activity of the ribosome binding site, making them difficult to describe in a standard way. And I bring this up because our iGEM team has a very talented undergraduate, Alec Nielsen, who has a poster that attempts to address this question. So if you have time, please check out his poster and give him some feedback. Biological systems are very complex at the level of molecular interactions. There was a recent review that came out by Eric Hazeltine and Francis Arnold in which they said details matter and we don't understand the details. This can sometimes stymie the attempt to make measurement standards. So given these complications, how realistic is it to develop accurate standards for measurement for biology? Some suggested goals of today's workshop, and these are by no means the only goals that we can have, but these are meant to function as talking points, to identify and define biological parts, to standardize types of biological parts, families, to discuss pros and cons of current proposed standardization methods as well as new characterization methods, to determine how to introduce groups and institutions to adopt standardization methods, to prioritize or at least discuss model organisms and conditions in which to perform measurements, and to develop realistic expectations for the achievement of measurement standards in the current state of the field, and then future goals for experimental measurement standards. And I'm just going to close my brief introduction with a quote that I heard Jason Morrison say, although I can't completely vouch that he made up this statement, but I heard him say, promote your standards and standardize your promoters. So I'm going to introduce today's speakers. These are the guys who are really knowledgeable about synthetic biology's measurement standards. Chris Anderson is going to talk about quantitative standards. Jason Kelly is going to discuss his promoter measurement kit. And Kim DeMora is going to talk about work he's done with yeast promoter expression levels.
There's kind of been one mindset of thinking about quantitative standards and how they're going to work, which a number of people, you mentioned Francis Arnold, have called into question whether that's a realistic model for how you would standardize or how you would communicate the activity of a part. And so just in the big picture, I want to talk about the various possibilities for how this could be done, and what the criteria were really for thinking about how you would pick one over another. So I'm not particularly advocating anything in particular. I'm just trying to set the framework for how one would distinguish things. So in general, what is the purpose of the standard? I mean, the whole goal in synthetic biology is to have this idea, an abstract idea in your mind about what the function of the system is that you want, and translate that into a DNA sequence that will impart that activity upon a cell. So the purpose of a standard in that context is so that you can encapsulate some piece of information about the function of a sequence of DNA and share that with someone, and it gives them some sort of predictive power about what it will do when they put that inside another sequence. So what is the sort of information you'd want to predict? You want to know ahead of time, is this composition going to give me the expression level I need? I think that's principally what we're concerned with. That's what we're usually thinking about. But there are other things about a part that are very useful to know. Often the failure of a composite part in making it in the lab is that it's toxic and you just can't make it, or you make it and the cells are really unhealthy. So that is additionally something you might want to try to standardize and communicate to somebody. Is this genetic circuit going to obey the kinetics that I've modeled? Then there are other things that are just simply biochemical in nature and not quantitative, just like, is this displace secretion composition going to display or secrete? That's something you'd want to try to encapsulate. So there are many models as to how you might go about doing this, and the first one is the one that most people advocate, and this is the one where you have absolute or relational standards. So here you have basic part things like promoters, ribosome binding sites, coding sequence, and terminators. And you're each stamping a number on them. that encapsulates units that are relevant to the type of a part. So this is the pops and the rips and the whatever. These numbers define the part's activity in physicochemical units in the case of an absolute measurement. So pops and rips were intended to be physicochemical measurements, or relational units to some sort of equivalent part in a standard composite. So there's some model of comparing the activity of this thing to something else that you know. All right, so this is an example of how you might use this sort of scenario for relational standards. So in one case, you've defined a standard composite, that thing in the top left corner, and you also know the activity of one perturbation of that sequence. So in this case, you replace the ribosome binding site with another, and you know the relative activity of those two things. So in this sort of model for standardization, the prediction you would make is that if this guy on the bottom is twice as active as the one on the top, then when you switch out the open reading frame, this guy is twice as active as the one on the top. So if you were going to have a method like this, your validation would look something like this. There's a 65% probability that this part with a standard composite score of 0.25 will have 25% of the activity observed with the cognate standard composite part within the area of measurement. So you want to be able to make some statement like that when you communicate the standard to people that's going to encapsulate what's going to happen when they use this part. So here's some real-world data speaking to this sort of type of modeling, and we'll hear more about it, I think, from Jason later. But this is a family of ribosome binding sites that we made in the lab, and we put this thing in two different contexts. We made the family with RFP and with LAC-Z. We measured their activity, and it looks like this. So when you look at the raw data, you find out that the rank order of things is roughly but not universally conserved. I mean, it's not always the case that the stronger promoters fall down, always being stronger than the weaker promoters. You do kind of find that the dynamic range, so like the ratio of the highest one to the lowest one, is roughly conserved for the whole family. Now, if you normalize this, which is in effect what you're doing when you say the composite part, so you pick one of them, you declare that to be the standard composite, and you take the ratio of all the activity to that family, what do you get? Well, you get 4 of them that match up, so that's good. You get 3 of them that don't. So in general, you get maybe 60% predictability out of this, which isn't horrible. You know, that's not a disaster, but one would hope that we could do a little better than that. So where does this break down? So we know of certain scenarios where this just doesn't work. So you can have composition or context effects that dictate expression levels more than the intrinsic rates of transcription, translation, or termination. So these can be secondary structure or occlusion of the ribosome binding sites, variation in mRNA processing or stability, anti-termination, supercoiling, you name it. There could be, there could be an siRNA sort of scenario in E. coli, like the thing we don't even know about that goes on in the bacteria that influences expression levels that we'll find out 10 years from now. We don't necessarily know that we know all the different things that affect expression levels. And here's an example of one of those weird things that just I can't explain. So we have the same PTET, RBS, RFP terminator basic part, and there are 6 different puck plasmids shown here. So they're all the same strain, they're all the same copy number. All that's changing is the context around the part. And you can see that it's like 5-fold times more active in one case than the other. So you can't blame that on secondary structure. You can't blame it on anything intrinsic to the parts. It has something to do with the context in the plasmids. So maybe it's the variation in supercoiling. Maybe it's the phasing, like the double-stranded nature of the DNA and where it is within the circle. Who knows? So you kind of have to wrestle with this if you're going to make predictability. So here's another model. So this is standard analysis. Here you have a standard set of algorithms. So the thing you're communicating here is not a number stamped to a part. It's an algorithm that you apply to the sequence of a device. Standard set of algorithms specific to a particular device type is applied to a composite part sequence. The product of the algorithm are theoretical biophysical parameters, which are then mapped to the phenotype data. So the Voigt lab has done one of these things, and but the general context of how you use it is you make one construct and you measure its phenotype. Then you apply algorithms that predict theoretical free energies of basic biophysical parameters. You then computationally predict a sequence that would have the desired free energy that you want for the activity of your final part, and then you make that construct, and there's going to be some prediction as to whether that thing is going to give the number you want. So your validation is going to look like this. For an algorithm-generated variance of initial sequence to be 25% of the original sequence's activity, there is an 85% probability that will be this activity within the area of measurement. That's the sort of statement you want to make about your algorithm. All right, the third is kind of the worst case scenario in that it gives you the least amount of prediction, but potentially it's actually faster than the other ones. But this is the idea of a standard set. So a standard set of composite parts that have a consistently observed probability of containing the desired activity set. Sets are defined for distinct device architectures. Okay, so this is not talking about libraries. So libraries would mean you make a bunch of random things. A standard set would mean you make these particular variants containing your part of interest, and there's some prediction about whether the activity you want is within that set. So this idea has been used multiple times within biology, but in one example it's been used in protein crystallization. So crystallization of proteins is one where you'd think the problem would be really hard. You're trying to determine the structure of the protein. Whether the thing makes crystals or not is a function of the structure of a protein, which is the thing you're trying to find out. So it seems like it's a fundamentally problematic scenario. It's been solved for about 15 years, mostly. There's certainly things, it's not a perfect field, but suffice it to say, what they do is you get this nice commercial kit from Hampton Research. You have 50 sets of reagents, and you set them all up in these crystallization trays, and then you're told how to analyze the data. You can even put that into a website that, based on the patterns you get, it will make predictions about what you should do next. And the bottom line is it's a design set that has been empirically made to contain some condition that is likely to have the acceptable conditions for your protein to crystallize. So although the probability that any particular one of these 50 conditions is likely to be the one that makes your protein crystallize, there's a fairly high probability that one of the 50 will be right. All right. So the sort of prediction you want to make in a standard set is that there's some high probability that a member of the standard set will contain the desired activity. So what would that mean in the context of an engineering scenario? So if you're talking about tuning the ribosome binding sites to get a particular level of activity, maybe it's you make these 12 ribosome binding site variants of your thing. So maybe 4 from one RBS library, 4 from another, and 4 from a third. And you make all those, and you just look at the activity of those 12 constructs, and there's some prediction that one member of this thing is going to have the right activity level. So in summary, the sequence-phenotype relationship in biology is complicated. We don't know that it's infinitely complicated, and we know it's not infinitely complicated. It's just complicated. The purpose of design standards is to minimize the time, cost, and uncertainty associated with identifying a sequence that confers a specific quantitative phenotype. Design standards are likely to involve a combination of forms with different approaches relevant for different scenarios, for different applications. So I'm not advocating that one of these three sort of approaches is the right one. It has to be empirically determined by using these sort of validation methods that give you some prediction as to how good of a prediction it makes as to which is the best way to approach each different scenario. Design standards must be validated in diverse cases. It is not sufficient to do one experiment and make a claim about a standard. Design standards require a theory of use. So within all of this, there has to be kind of a whole theory based on lots of data that tells you why you're doing certain things and how you approach the problem and pick the standardization method that's right for your application. All right? You up next? Yeah.
For your RFP and LAX data,
Yeah.
is the rank order conserved when you look between like steady state measurements and dynamic measurements? Do you know?
Steady state versus dynamic. What does that even mean?
Oh, just like I'm just curious if you measured it in the growth phase versus stationary phase, basically.
So these are mid-log.
These are mid-log, okay.
Yeah.
I'm just curious if it would change at stationary phase. I don't know.
My guess is in this case it probably wouldn't, but I mean you definitely, in other experiment scenarios, like when we did the AND gate, there were certain pieces of that system that had a very clear OD dependence. So you would get different results if you looked at it at different ODs. Right. In this case, I don't think it would. Yeah. I mean, you saw the pattern of activity is apparent in colonies, it's apparent in midlog, it's apparent in the saturated cultures. I don't think the rank order matters.
Okay.